All right, next up, we're going to do a couple of sections on transport in plants, looking at both movement of water as well as carbohydrates throughout the body of a plant. It's kind of the last little section of our chapter here. Um, the overall pathway, we're going to start off with water first, I guess, first of all. We're going to start off with water. And so when we're looking at water moving into and through a plant, water is going to come in through the roots, and as we'll see in particular, that's going to involve the root hairs. It's then going to move into the xylem through one of about 3 different routes the water can go into the xylem, which of course we know is vascular tissue and is then equipped to take water up through the rest of the plant. Now, the tricky part about this, or the part that people didn't understand for a long time, I shouldn't really say it's tricky because it's not that convoluted, but the water has to be able to go up the xylem. Right? And so basically we have water coming in through the roots, going against the force of gravity, sometimes upwards of 300 or 350 feet for a really tall tree, like for example out in the western U.S. That's a really long ways to go against the force of gravity. And so understanding how water is able to rise over that distance is a really important part of this section of this chapter. Eventually, water is going to leave through the stomata in the leaves, from those little mouth-like openings in the leaves, from which water evaporates, and thereby completing the process of water movement. And so that evaporation of water, or what we call transpiration, we talked about that, I believe, in our last section or 2 sections ago, is really instrumental in making sure water moves up from the roots through the stem and eventually to the leaves. Now, as far as water coming into the roots, there is a very, very slight amount of water, in a way, sort of pushing its way into the roots. The tissues inside the roots have a bit of a degree of solute concentration, so water and so forth are going to tend to come into the roots on their own, but that's a relatively minimal force. Most of the water movement in the plant is actually going to come from the pulling of water that results from transpiration or evaporation of water at the leaves. That's where 90 or 95% of the force of water being moved through the tree or through the plant is going to take place. Is the pulling of water, not the pushing of water. And so in particular here we're talking about evaporation of water from the stomata. Now, when that one molecule of water leaves, and you think about this one little molecule of water leaving from the stomata up here goes water up here. That water molecule is connected to every other water molecule lying behind it. Water is a very unique molecule. It's got some special properties. We talk about cohesion of water molecules, so water molecules tend to stick to each other. And we also talk about adhesion of water molecules. So not only are the water molecules sticking to each other, they're also sticking to the sides of the tubes. The sides of these xylem tubes are made out of cellulose. It's got some similar chemical properties to water, so water and cellulose get along pretty well, and that allows the water to also stick to the sides of the tube so it doesn't fall back down with the force of gravity. So the evaporating of water molecules from the stomata, in addition to cohesion and adhesion of water molecules, allows the water to go from the roots up the stem and eventually out through the leaves. Now, as far as getting water into the xylem in particular, there are a couple of things we have to look at here. Water molecules typically, as well as other things, ions and so forth, are going to have to use protein transporters that are built into the cell membrane. We talked about cells and cell membranes before, and we talked just briefly about transport processes. So facilitated diffusion and active transport and those kinds of things in particular are going to be really important. So sort of looking over here at this picture, we have these protein channels that are equipped to move, in some cases, what are called symporters. These are moving a molecule like sucrose and a hydrogen ion in the same direction from outside the cell to inside the cell. So it's basically facilitated diffusion of 2 things at the same time. Sucrose, which is just table sugar, and hydrogen ions are moving in the same direction through this protein transporter. Over here we have an ion channel. So we talked about channel proteins way back. Channel proteins are going to allow, in this case, potassium ions to come right through this transport channel into the cell. Simple osmosis and diffusion, on the other hand, these things go right through the phospholipid bilayer, and most of the things we're dealing with in this chapter are not going to move that way. We're going to see some channels, we're going to see some symporters, and we're going to see some pumps that are involved in moving things against a concentration gradient, in the process also using ATP at the same time. Because remember, when you're going against the concentration gradient, going from low concentration to high concentration, in this case from the inside to the outside, that's going to require some energy, or specifically it's going to require some ATP. So we call those hydrogen pumps or proton pumps. They're going to help us move H pluses against a concentration gradient, thereby allowing for some really important processes to happen when it comes to water movement. Remember with osmosis, we talked about this as well. Osmosis is where you have water going from a less concentrated solution to a more concentrated solution in terms of the solutes, that's going to be the general overall direction of water movement. That's also going to be really important for this section of this chapter. So we're not actually going to get into the math of water potential, but just like we talked about potential energy, there's also potential as it pertains to water movement. So when you think about having a high water potential, think about that as being a way to store up potential energy in a way. And so we have this overall idea of water potential. We use this funny little Greek letter psi with a subscript W to represent overall water potential. And so the main thing you want to remember, and again, we're not getting into calculations, we're not even really going to look too much at the formula here, but the main thing you want to remember is that water always goes from where there's a high water potential to a low water potential. It always moves in that direction, just like things would tend to move from the top of a staircase to the bottom of a staircase. Think about that as being water potential. It's going from where it's higher to where it's lower. Okay, so if you take a plant cell, for example, it's a plain old plant cell, stick it into plain fresh water, pure fresh water, water is going to tend to rush into that plant cell because there's more stuff dissolved in the plant cell, in the cytoplasm, than there is stuff dissolved out there in that pure water. So water is going to tend to come in. Another way of saying that is the solution would be hypotonic and the interior of the plant cell would be hypertonic. So water is going to enter the cell. Since solutes are higher inside the cell than outside, water moves in through osmosis. And because the plant cell has a cell wall, we know it's not going to burst. We know it's relatively stable. We know it's not going to explode because of water rushing in. The cell wall is going to help contain water rushing in, and the cell is going to become turgid, and that's really where a plant cell wants to be. On the other hand, if you were to place a cell in a really concentrated solution of sucrose or really concentrated solution of salt water, that would be more of a hypertonic solution. The plant cells, therefore, would be hypotonic, and water would tend to leave the plant cell in that case. Okay, so we look at water potential in terms of two main forces here. There are physical forces involved, like the plant cell wall, which helps to push water out if water is coming in, as well as gravity. All right, so since gravity is a relatively minimal force, we're going to kind of ignore that one and focus primarily on the physical force of the cell wall versus the solute potential of the cell. Okay, so we look at what's called pressure potential. That's this Greek letter psi with a subscript of a p, pressure potential, and we also look at something down here called solute potential. Right, so it's really pretty much that straightforward. We've got a total overall water potential where water always goes from the higher to the lower side. Usually that means going from less negative to becoming more negative in terms of water potential. We can look at the physical forces involved, primarily that of the plant cell wall, and we can also look at the chemical forces involved with the concentration of solute into solutions on either side of a membrane. All right, so if we take a look here at a plant cell, right, we've got a plant cell with a really cool, apparently rooster pot that's growing in, I guess. We've got a plant cell. We've got some solution into which the plant cell has been suspended. And in this plant cell, there are relatively few solute molecules. There are a lot of solute molecules outside the plant cell. So in this case, because this plant cell does not really have much in the way of a cell wall, the solution doesn't have much of a cell wall, I misspoke. The solution doesn't have much of a cell wall. We're going to assign the solute potential a value of negative 0.7, and that's just based on how many of these little dots are floating around here in this water. The pressure potential is 0 because there's no cell wall surrounding this to offer force back in the opposite direction. So the overall water potential is negative 0.7 megapascals, which is a unit of pressure. Okay. If we look at the plant cell now has a solute potential of negative 0.2. It's not as negative as this because there are fewer solute molecules floating around inside of there, so it's a less negative number. And the pressure potential is going to be a positive 0.5 because the cell wall has a wall pushing water out. If water rushes in, the cell wall pushes back in kind of the opposite direction. So if we look at the balance here, we have a negative 0.2 because of solute potential. We have a positive 0.5 because of pressure potential. That equals out to 0.3 positive megapascals units worth of pressure. So we need to think now about what happens in terms of overall water potential, the overall water potential of the cell in solution and where exactly water is going to go. Okay, what did we say? What is our rule? Water always goes from a higher number to a lower number. It goes from where there's a higher pressure to a lower pressure. So if the inside of the cell has an overall pressure of 0.3, the outside of the cell has an overall pressure of negative 0.7, water is going to go from inside the cell, where it's plus 0.3, to outside the cell, where it's negative 0.7. Water always goes from where there's more to where there's less. And that's why osmosis is the way that it is. It's why it works the way that it does, is because of these pressures involved. But now we're actually assigning physical forces. In this case here, this cell put into this solution initially is going to tend to lose water. And you know that just by looking at it. I mean, there's more water proportionally inside the cell than outside the cell, so obviously water is going to tend to leave. And that's exactly what happens because of pressure potential. Eventually the cell is going to shrivel up. This cell here is going through plasmolysis. The cell membrane is actually shrinking away from the cell wall because it's lost so much water to the outside environment, it's going to shrivel up and the overall plant itself is going to wilt. Eventually what happens is that the cell water potential and the solution's water potential are both at negative 0.7, and that plant cell is not especially happy in that case. All right, so we mentioned before root hairs. Root hairs, remember, are epidermal cells on the roots that actually do most of the work when it comes to absorption of things, whether it's water or ions or both. We also see an association called a mycorrhizal fungal association here. Mycorrhiza literally means fungus root. And so in and around these root hair cells are mycorrhizal fungi that colonize the roots and also greatly help to increase the surface area of those roots to help facilitate further water transport and really help these plants get more water. Some other things mycorrhizal fungi do, they're also involved in taking up phosphorus, which is something most plants are limited by. So phosphorus uptake is enhanced by having these fungal associations. And there's also protection here. So because the plant cells are colonized by these fungi, there's less opportunity for other nastier pathogenic type fungi to come in and attack the roots because they're already being kind of protected by these mycorrhizal fungi. So it's really kind of a threefold benefit for the plant. The plant's getting protection, it's getting phosphorus, it's getting extra water, and the fungus is getting a place to live, and it's getting carbohydrates from the plant. So this is a type of relationship that we call a mutualism, where both parties are going to benefit. Both species are benefited by the presence of the other one. It's a win-win association, so we call that a mutualism, and that's something we're going to see more once we get into our chapter on community ecology. But in any case, having these root hairs and having these mycorrhizal fungi help to increase surface area, thereby increasing uptake of all substances by those roots. This graph here, we see an overall picture of the plant as a whole in terms of water potential, so starting off in the soil, moving into the roots, eventually up the stem to the leaves, and then eventually into the air. Water potential gets more and more and more negative, so negative 0.5, negative 0.1, eventually as much as negative 100 megapascals in the air outside the plant. Very dry air would have to be, in that case, to have that negative of water potential. That's why water moves the way that it moves. Over here in this picture, we see a more microscopic view, a cellular view of what's actually going on now. So we have soil particles surrounded by water. We have a root hair extension coming out from the root itself, and we're going to have water coming in through that root hair, eventually making its way into the xylem, and eventually the xylem is going to carry that water up into the leaves. The other thing that's happening over here is what's known as a symporter. And so these symporters, again, are proteins built into the membranes of cells that are equipped to move two different substances in the same direction. Sym means together, and porter refers to transporter. So they're moving, in this case, both hydrogen ions and mineral ions in the same direction. Right? So this is a case of facilitated transport. In this case, this one protein is actually moving two things at once from outside the cell to inside the cell. And what you also want to think about here is because we're bringing in H pluses, because we're bringing in minerals, what is that going to do to the tendency of water? Where's water going to tend to want to go if all of a sudden the inside of the cell is full of these solutes? Water is going to want to go into the cell as well. So this is another way to help maximize uptake of water is by having this continual uptake of ions and minerals into the cells themselves. All right, so we look again at the same picture from the last slide with our tree. We look at what's happening up in the leaves with these stomata. Right, so we have, in this case, water exiting from the stomata. Evaporation is occurring, and because these water molecules are attracted not only to each other, these are water molecules here. So these white structures here are hydrogens. This red one in the middle is an oxygen. They're attracted not only to each other through what are called hydrogen bonds, but they're also attracted to the walls of the xylem tubes, which are made of cellulose, and that's going to give them some reinforcement as well as they're being pulled up through that plant. I mentioned before, early on in this section of the lecture, how there are different mechanisms for water to get through the cells in the roots, eventually making their way into the xylem. There are three main routes of transport, and so what you want to think about for each one of these is, first off, how quickly it occurs, but secondly, how much control the plant has over where the water actually goes and what might actually be in the water. So how long does it take, as well as what's actually being moved in that water as it's coming into the plant. So the first type of transport is called apoplastic transport. And apoplastic transport is where the water goes around the cell walls. It actually goes through the cell walls and doesn't go through any plasma membranes or cell membranes of any kind. Right? It's relatively quick, it's fast, but plants do not have a great degree of control over what gets to enter into the xylem. Whatever's in that water is going to go into the xylem automatically as well. So that may or may not be a good thing. The second route is called symplastic transport. So if we look at this green arrow, we have water coming in from the outside environment through one plasma membrane, through a second plasma membrane, to a third and fourth and fifth and sixth plasma membrane. If the xylem is over here, that water has had to pass through 6 different plasma membranes that have all had an opportunity to filter out things that might be in that water that maybe the plant doesn't want in the xylem. It's not as fast as apoplastic, but it also offers a great deal more control over what actually goes into the xylem. And the third route of transport is called transmembrane transport, and this one's going to go through several more membranes on its way to get into the xylem. So now we have not only plasma membrane 1, we have vacuole membrane 1, vacuole membrane 2, plasma membrane 2 and 3, vacuole membrane 3 and 4, plasma membrane 4 and 5, vacuole membrane 5 and 6, and then plasma membrane 6. So we're actually going through 12 different membranes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and there's number 12 right there. So obviously this is going to take a lot longer because it has to actually physically get through these membranes each time it encounters one. It's going to take a while, but it also gives the plants the most control over what actually gets into the xylem. All right, the other kind of filtering aspect, the last line of defense before water actually gets into the xylem, is that Casparian strip that we talked about back in one of our earlier sections. And remember we talked about the endodermis, the endodermal cells that surround the stele and surround the xylem tissue are surrounded and permeated with this Casparian strip, which is basically a layer of wax that forces water to go through the endodermal cells as opposed to going around the endodermal cells. So again, because water is eventually going into the xylem and from there on to the rest of the plant, it's really a good idea to have a last final filter here for that water. Before any water gets in through any of these mechanisms, apo, symplastic, or transmembrane movement of water, regardless of which one that goes through, all water eventually has to go through the endodermal cells because it can't go around them due to the Casparian strip. So that's kind of our last layer of control over what gets into the xylem, and then from there moves up to the rest of the plant. If we look at mineral ions, typically in the soil, minerals now, not other things, but minerals have a higher concentration there than in the plant itself. And so if we're going to go into the plant, right, if we're going to accumulate ions into the plant cells, we're going to have to work on spending some energy, right? So we have this high concentration of ions and minerals inside the plant. We have a lower concentration outside the plant. And so we're going to have to spend some energy to move against our concentration gradient. We're going against a concentration gradient from low concentration outside to high concentration inside the plant. And of course, to do that, to go against the concentration gradient, we're going to have to spend some ATP. So we're doing active transport here. So what's going to occur here is that in addition to the active transport that goes on at the level of the plasma membrane, especially with root hair cells, there are also protein transporters involved with the endodermal cells to help move some of these ions into the xylem so they can eventually make their way into the rest of the plant. And so these are also known as proton pumps. We're moving very specific ions against the concentration gradients more and more and more as we approach the xylem until eventually they get into those xylem transporters themselves. All right, so we're going to stop here. I'm going to cut off and I'll start up our very last section of our lecture on plant form.